Hello everyone, happy to meet you all in yet another video. Let us discuss about the pathogenic bacteria Coxiella burnetii. First, general characteristics followed by transmission, pathogenicity and the clinical disease, treatment and prevention. Now let us discuss about the general characteristics of Coxiella burnetii. In gram staining, it shows gram-negative rod-shaped bacteria arrangement. It is arranged in the form of Coccobacillus and motility. It is a non-motile bacterium. Capsule is absent. Endospores is also absent. Respiration, aerobic and facultative anaerobe. Aerobic, which requires oxygen for growth. Facultative anaerobe, which does not require oxygen for growth, but it also grows well in its presence. Temperatures, it requires 37 degrees Celsius. Optimum pH, it requires 4.7 to 5.7. Burner discovered the bacterium Coxiella burnetii. Later it is named in his honor, which causes a fatal Q fever. And next, pathogenicity of Coxiella burnetii. Disease transmission. This bacteria would be transmitted via the inhalation of aerosols. This may also infect the person who work in the farms and also in the tanneries. And next, incubation period. It may develop in 2 to 3 weeks or it may take 2 to 6 weeks. Virulence factor. There are two virulence factors for Coxiella burnetii, and one is phagolysosome and another one is lipopolysaccharide. And now let us move on to the pathogenesis of Coxiella burnetii. The infection may occur through the inhalation of aerosols in the scent which contains the bacteria. Ingestion of contaminated dairy products can also be the source of infection. Once the bacteria enters the host, the bacteria is phagocytosized by the macrophage. It can resist the host condition of the phagolysosome. They will replicate inside the phagolysosome. After the multiplication or replication, the bacteria with this mechanism evade the host immune system, then are capable to establish the persistent infection. The bacterium can disseminate systemically, which affects the various organs such as livers, lungs, and spleen. Clinical disease caused by Coxiella burnetii in that acute Q fever, flu-like symptoms which causes the fever, headache, muscle ache and fatigue. And then pneumonia is a severe clinical condition which gives us a respiratory symptoms like difficulty breathing, cough, etc. And next, hepatitis is a condition which causes inflammation of the liver. Chronic Q fever, it is also called as endocarditis, which is the inflammation of the inside lining of the heart chamber and heart valves. Now let us discuss how Coxiella burnetii is diagnosed under laboratory. Microscopic examination. In gram staining, it will appear as pink colored gram negative rod shaped bacteria. In motility, they are observed as non-motile bacterium. Colony morphology on culture medium. Selective medium used for the Coxiella burnetii is acidified citrate cystine medium, which will gives a ghost-like appearance with bacteria staining, contains chromatin but also in the form of devoid of cytoplasmic content. Biochemical test. Coxiella burnetii can't be diagnosed using biochemical test because it does not show any metabolic activities like other bacteria. There are other certain methods followed to diagnose the Coxiella burnetii. In serological test, immunofluorescence assay, enzyme-linked immunosorbent assay is used to diagnose Coxiella burnetii. In molecular methods, polymerase chain reaction is used to detect the Coxiella burnetii. Culture, the bacterium Coxiella burnetii is cultivated in a special cell culture technique where it can be used for various study purposes. Treatment, the antibiotic doxycycline is used for the treatment of Q fever as both in adults and children. In pregnant women, it is suggested to take an antibiotic like a cotrimoxazole. Prevention, it is suggested to avoid contact with the infected animals and also do not consume the contaminated milk products. Thank you.